Hi guys, and welcome to HSC Chemistry and the acidic environment video series. We're going to continue on with our look at titration, specifically look at the technique of volumetric analysis, specifically through titration. So volumetric analysis is one of the important techniques in chemistry, and we contrast volumetric analysis with the other one that we've had a look at before, which is gravimetric analysis. A gravimetric is based on mass, comparisons based on the mass of components in a mixture, for example. Volumetric analysis is based on concentrations of solutions. So we're trying to find unknown concentrations of acids and bases. Now, the technique of volumetric analysis can be applied to things other than just neutralization reactions, and you may see a couple of examples of these later in the course. But what we're going to be doing in this particular, or looking at least in this particular video and duplicating in class, is going to be the technique of volumetric analysis known as titration. Volumetric analysis is used to measure the volume of a solution of known concentration needed to react with a solution of unknown concentration. So we have to have something that's known in order to calculate something that is unknown. The key to this is something called the point of equivalence, which is what we're looking for in our neutralization reactions. I mean, ideally, we would like to pick the point when the acid and base neutralize each other to produce a pH of 7. That's theoretically what we would like to do. In reality, that is not always what happens, and we'll see some examples of why that's the case as we continue on. So equivalence point really is, I guess to define it, it's the point where the amounts of each reactant in a chemical reaction are sufficient to cause complete reaction using both reactants. So we are not talking about limiting agents. Okay, we don't want limiting agents, and we don't want agents in excess. Okay, both of these are not what we want. We want to try and find the point as much as possible where we have a complete reaction. Complete reaction. That's what we're after in this particular case. Now, two important, I guess, concepts that we need to just emphasize as we're going through our titrations is the difference between an equivalence point and an end point. Sometimes these are the same and sometimes they are not. The equivalence point is ideally most of the time where the pH is around 7. So in an acid-base reaction, if the pH was 7, that would be the equivalence point. But we may not necessarily be able to stop our reaction at exactly a pH of 7. It may slightly overshoot that mark. And that's one of the problems, in that sometimes the endpoint is a little bit beyond where our equivalence is. So we try and make sure that they're as close as possible to one another. The procedure for a titration then is all about setting up a burette which has a solution of acid or base in there. The opposite is what will be in the conical flask. Conical flask, this is the burette. And of course, in there is also going to be an indicator. And the particular indicator chosen is going to be one that helps us to identify that end point, hopefully the equivalence point, or at least as close as possible to that when we reach the point where we stop the titration. One of the keys of this procedure is trying to get that point as close as possible. You can see in the example that I've given you, this is using the indicator phenolphthalein. Now, phenolphthalein is an indicator which is colorless. I'm sure you remember, below pH of around 8, but turns to a reddy pink or magenta color once it gets to around about, well, over 8 to 10 and beyond. It just deepens in its pink color. What that means is that this one here is a really bad titration because it's massively overshot, so it's an overshoot. Okay, and that can happen with a couple of drops. It can happen with a single drop. That's how precise this technique is. This, on the other hand, is a fantastic color. It's the sort of color that we want. You can see there's just that tinge of pink in there. Maybe even less than that would be even better, but at least we're trying to get a color where we just see that pink color appear, and certainly not something that is massively overshot like the second one. When we're titrating, there are 4 types of reactions that involve acids and bases. One of those types of reactions we never do. If you notice from the list that I've given you, the one that you should see is missing is weak acid with weak base. Fact of the matter is both of these by definition are not fully ionized, and therefore we have to keep shifting the equilibrium. The process is so slow that it's just not worth doing, and we just don't titrate weak acids and weak bases together, so we don't have to worry about that one. So therefore we've got really only 3 that we need to deal with: a strong acid with a strong base, a weak acid with a strong base, and a strong acid with a weak base. Now I'm going to suggest that one of the best indicators for anything that involves a strong base is phenolphthalein. Oops. It's a shocking word to try and spell. Phenolphthalein. And that is likewise the case for a weak acid and a strong base, phenolphthalein. There's reasons why this happens, and we'll actually have a look at some of these as we get into class. And the only time that I would not use phenolphthalein is when I have a weak base. When I have a weak base, that's when I'll use something like methyl orange. This has to do with the nature of the salts that are being formed. So a weak base is going to form a strong conjugate acid, and this strong conjugate acid is going to drop the pH of the end point, which means methyl orange is actually the best choice in this case. The strong base will have weak acid conjugates, and as a result, something that's changing in a pH range of 8 to 10 is perfect for these 2 types of titrations. We're going to be carrying these out in class during this week, so I'll expand on a few of these when I see you then. Thanks for watching.